Okay, so I'm pleased to welcome today's seminar speaker, Dr. Andre Naranjo. Andre is the botanical curator at Florida International University. He received his Bachelor of Science from the University of Miami and his PhD from the University of Florida, working in the Soltis Lab. He was an NSF graduate research fellow and later an NSF postdoc, also at UM. So as we'll hear today, his research focuses on phylogenetics and biogeography. And though I'll be a bit biased, I consider his work to be some of the most exciting phylogenetics research going on in our region. As he has plenty of Dicerandra, Conradina, and in his talk. And he is no stranger to the scrub, having spent quite a bit of time working with scrub mints here and all over the Southeast. So lastly, I'll just note that Andre will be starting a new position at FIU as a research scientist and curator of a new herbarium and the curator of the herbarium and living collections at FIU, which is a rare thing to hear of a new herbarium. Andre will be heading it up. So let's welcome Andre.
Thank you for that amazing intro, Aaron. I really appreciate it. So yeah, I'm here essentially to talk more or less about work that I did during my doctoral dissertation, working in the Soltis lab. Granted, this is work that I still continue to kind of work on, and I'm going to continue publishing more things. And just had a meeting today with Aaron and Ray Crandall over at UF to potentially begin some sort of Dicerandra working group of some sort, so that we can really kind of begin to focus and hone our efforts to protecting and saving these really charismatic little mints that we find throughout the Southeast. All right. So to start off, we obviously have to give some context as to where we are, right? The North American coastal plain was described in 2015 by Reed Noss as a global biodiversity hotspot. Most of its modern ecosystems arose during the late Pliocene to the early Pleistocene, so about 6 to 4 to up to 2.5 million years ago. It's home to over 1,800 endemic vascular plant species, so that's pretty remarkable in and of itself, especially for an area that has such low topographic relief. There's a lot of homogeneity in terms of elevation across it, right? And I guess most tragically is that over 70% of the original habitat that made it up is now gone, mostly longleaf pine forests, right? So within the North American coastal plain, there is a clade, or a group that shares one common ancestor, that we like to informally, I guess, call the scrub mint clade. It's made up of about 24 different species that are, strictly speaking, endemic entirely to this biodiversity hotspot. The largest of the genera that make up this clade is Dicerandra. There might be a bit, I admit in advance, a bit of a Dicerandra bias in this talk, partially because I think that there are a lot of very interesting and compelling questions within the group because of their different life histories and styles, and just the breadth of range differences that they have, right? So Oliveira et al., together with Pam and Doug Soltis back in 2006, created a very preliminary phylogeny of the group using several different genetic markers, and they were able to confirm the existence of these two different subclades within Dicerandra, the perennial and the annual subclades. Perennials have, as you might know, very restricted distributions along these scrub or ancient ridge refugia, either along the Lake Wales Ridge here in Central Florida or along the Atlantic Coastal Ridge. Annual species, generally speaking, have wider distributions further north. They're mostly found north of the Gainesville area. They tend to prefer slightly less scrub and xeric habitats. They usually occupy sandhills. Many of them occur along streams, in the case of Odoratissima and Radfordiana, which I'll touch upon in a little bit. But in general, annuals are much more widely distributed. There's examples of hybridization occurring between sympatric ranges where two different annual species might run into each other. And I guess the only exception with wider ranges is this Dicerandra Radfordiana, which was described by Robin Huck, who was the one that the individual that did most of the taxonomic descriptions for members of this group. But that one's a bit of an oddball, and many other scientists will occasionally consider it as just a morph of Dicerandra odoratissimum. The next largest genus in the group is Conradina. It's similar to Dicerandra, or at least perennial Dicerandra, in the sense that they're all made up of these relatively short-lived but woody perennial subshrubs. They're found in several different areas, either along the Atlantic coastal ridge in the case of grandiflora. Brevifolia occurs here on the Lake Wales Ridge. Conradina edenaya and signiflora are two very closely related species that are found in these very small state parks in, I believe that's Putnam County. Canessus and glabra are kind of found in the Apalachicola area. And perhaps the oddball of the group is Conradina verticillata, which is unique in several different regards. As you might be able to tell, it's not technically in the coastal plain. However, Story and Weekly back in 2001 described the Cumberland Plateau as a well-described refugia for a lot of coastal plain endemics, and might have acted as some sort of museum or nursery of biodiversity of coastal plain endemic plants during the Pleistocene. It has also a pretty different life history than the other ones in the sense that it is a perennial, but it usually is found either directly on stream banks. Sometimes it's even found in running water. So very different kind of sort of ecological niche compared to its more scrubby and sandhill relatives further south. And then we have this little bit of a mess in these other described species that fall within the scrub mint clade. We have two monotypic genera. There's Pyloblephus rigida. It's a species that's found almost entirely, it's endemic to peninsular Florida. It's a pretty good landscape plant, so I highly recommend planting Pyloblephus whenever you can. It smells amazing, and it's a great pollinator attractor. And then we have Stachydioma graveolens, which is an Apalachicola endemic. Then what we have is this kind of grade. To talk about clades, grade is perhaps the opposite. It's these plants that were described as all belonging to Clinopodium, which is a well-known junk genus for a lot of mints, both in Eastern North America and elsewhere. But of those Clinopodiums that are traditionally included within the scrub mint clade, we have Clinopodium georgianum, which is the one with the widest range out of all the scrub mints. And then the more narrowly, oddly distributed Clinopodium dentatum, which is endemic to Apalachicola. Ashii, which is described from these different populations, disjunct populations either along the Lake Wales Ridge, like we were seeing today, a little bit further north, and even all the way up into the Altamaha River Valley area. And then we have Clinopodium coccineum, which is traditionally referred to as just one species. Lately, there's been a little bit of talk about perhaps considering the Central Florida populations of coccineum as macrocalyx. However, I didn't go too deep into sampling for this group. I just wanted to have one representative from each of these Clinopodium species to more or less see where they fall in the tree, right? So I guess the general questions that I was kind of going into this whole project with were pretty simple. First, how did hybridization affect diversification in this clade, right? In what areas did the ancestors of these living scrub mint species exist? And subsequently, what kind of endemism hotspots can we identify today from looking at these plants? And are these hotspots more a result of neo or paleoendemism? So ultimately, the goals are to use the scrub mints as a sort of model for understanding biogeographic patterns and evolution across this biodiversity hotspot, and then to also understand the range evolution of plants that are made up of both more widely distributed and more narrowly distributed species, right? Within the context, again, of a biodiversity hotspot. So as I mentioned before, in 2006, some preliminary work was done on Dicerandra and likewise on Conradina as well, but again, a little bit of a Dicerandra bias here. And we were, this work was able to identify with pretty strong support that the perennials and annual Dicerandras do form these two distinct clades that are monophyletic and that have some pretty good bootstrap support as well. So this is a maximum likelihood tree if you're familiar with the methods. But as you can tell, relationships within each were poorly resolved. Occasionally there's these combs, there's low support values, there's a whole bunch of different things going on here. Kind of make it difficult to understand how each species relates to its congener, right? So in order to be able to answer these questions, we needed a better tree. So that's what I did. So I utilized something called a target capture hybrid seek. There's a whole bunch of different names for it. But essentially what I did was I used transcriptomes or protein coding segments of the genome of 18 different species of Nepetioid mints to identify a suite of different genes that I could then amplify and sequence in order to understand evolution of this group, right, using nuclear loci. So in total, I got 238 nuclear genes that I was able to sequence. I built gene trees for each of those individual genes. And then using something called a multi-species coalescent method implemented in this program called Astral, I was able to build a species tree. The species tree, so the funny thing, and I'll talk about this a little bit more later. So even though, the species tree that I'm showing here, it's a dated species tree, had very high support for almost all of the nodes. So it indicates that there's some sort of coalescent signal that the program is able to pick up on. It's like, okay, yeah, these are probably true distinct species. But individual gene tree support for these different clades was relative. So that's just something to keep in mind. I'm going to talk about that a little bit more in the next slide. But this is our dated analysis of using that same species tree. There are no fossils in Dicerandra, and there are even fewer fossils in the subfamily. These mints, they don't really tend to preserve very well. So what I had to do was use a palynological records, this little pollen grain of salvia, as a secondary calibration for my tree. So I use Monarda punctata as an outgroup, very close related to the entire clade as well. But just to generally talk about ages here in this group, the scrub mint clade ancestor occurred approximately about 4 million years ago during the late Pliocene. Dicerandra, so late Pliocene is more or less right when we begin to see some glacial cycling, right? There seems to be these colder, warmer periods, these drier, wetter periods as well, right? With the beginning of the Pleistocene, which corresponds almost perfectly with the beginning of Dicerandra, or rather this reported ancestral date for the ancestor of all of Dicerandra, about 2.4 million years ago. That's right, more or less when the Pleistocene begins. Conradina found slightly, more or less the same age as well. So 2.39 for Dicerandra, 2.48 for Conradina. So those are a few things. And then, like there's already pretty strong support for it. Was able to recover both the annual clade and the perennial clade of Dicerandra with pretty strong support. And each one of those started diversifying relatively shortly after this most recent common ancestor for all of Dicerandra, so about 2 million years ago. So I really do like to say that these scrub mints, particularly Dicerandra, are like truly children of the Pleistocene. It was the confluence of factors that the Pleistocene was able to afford them, and their other southeastern mint species really allowed them to kind of expand their range, enter suitable habitat, colonize new habitats that were emerging from the sea at the time, wink, wink, Lake Wells Ridge. And so it's through that I kind of wanted to really look at, okay, so we know it's the Pleistocene, something's going on. Exactly what other mechanisms are at play genetically and biogeographically and climatologically that's pushing this diversification that we see, right? So I threw the whole kitchen sink at my genetic data set. I was really interested, I was looking at these gene trees, I was looking at these species trees. I was looking at how few gene trees were supporting these different nodes within our species tree. I even talked to the authors of the multi-species coalescent method, and they were like, "No, if you're getting good posterior support for all of these different nodes in your species tree, but your gene trees are bad, don't worry about it. We take that into consideration in the algorithm that we use." I'm like, okay. So let me explain more or less why these gene trees are so discordant. There are several different things I wanted to look at. So on the upper left hand is something called the reticulation index, recently developed by a postdoc at Harvard. Essentially looks at all of these different sequences, looks at gene trees and looks at species trees and does a variety of different calculations in order to determine just the level of reticulation or hybridization. It's just another fancy way of saying it, within each of these nodes across the phylogeny. I also wanted to look at a gene concordance factor, which is similar to gene tree and species tree concordance. So the higher the value, the better the concordance is between gene trees and species trees. The lower the value, the lower, right? Occasionally, gene tree estimation error is a bit of an issue in phylogenetics and phylogenomics in particular with these large data sets. So I wanted to at least discard maybe, maybe the discordance that we see between these two different data sets is caused by estimation error. And then I also wanted to look at something called introgression fraction, which is essentially just the percentage of loci in a particular species that is identical to that of another species. To essentially be able to determine percentage of the genome or of these genes that might have been introgressed, meaning passed from one species to another during a hybridization. Yeah. Within the nodes, within the nodes. So this is like we're not looking necessarily at extant things, we're looking at things in the ancient past. So we're not looking at hybrids as we would think of them nowadays. Like we have a common garden, we have two plants next to each other, they produce seed that is hybrid origin, and that plant is inherently different. We're talking about hybridization hundreds of thousands of years ago, right? So all within this clade, right? So I guess several different things to look at. First off, gene concordance is really low. Like I mentioned before, gene trees don't really correspond very well to the species tree, even though the species tree has high support. So that's one thing. The next thing I wanted to look at was gene tree estimation error. And so there are some areas where there's very low estimation error. So if there's a 100, it means that estimation error was low. If there is a lighter blue color, then that means that estimation error was high. But the thing about estimation error is that it is directly correlated with introgression. So the more hybridization you find in a clade or in a node, the more likely you are to have higher estimation error, right? That kind of makes sense intuitively. So I guess the two ones I really wanted to highlight here are reticulation index and introgression fraction. Let me make sure this is So first off, when we look at the reticulation index tree, so blue values indicate low reticulation indices, and then obviously red means high. The hottest hotspot on the entire tree is the backbone of perennial Dicerange. I guess unsurprisingly. I guess one of the things that I was expecting when I was doing my analyses was a kind of a clade-based grouping of Atlantic coastal species with lake whales endemics, right? They'd be in different clades, they'd be related to one another. But actually what I saw was this sort of stepwise fashion where there was like this Christmanii, Cornutissima, then Thenicola, then Fructescens, then Modesta, then Immaculata. Pretty interesting stuff. So what we know, based off of RI, is we're able to infer that there was a relatively high amount of reticulation going on across the ancient history of these Dicerangia perennials. Likewise, some pretty high values in the annuals as well, but the highest values in the entire tree were along perennial Dicerandra. When we look at introgression fraction, which doesn't necessarily, it measures similar things, but in this case, it's looking directly at percentage of loci that have a hybrid origin. And we look at the heat map, and this area in particular stands out to me. All of the perennial Dicerandra have higher introgression fractions than you would expect just due to random chance. So again, corroborated by two different metrics, there is probably the diversity that we see in perennial Dicerandra is very likely shaped by hybridization. All right. A few other, then I guess the next interesting thing that kind of came out in these results. I had mentioned before that, actually I didn't mention. So these clinopodiums that I had mentioned before, this sort of junk genus that a lot of these scrub mints have gotten placed into, 3 of them formed a clade. And interestingly enough, the 3 that formed a clade were Coccineum, Aschii, and Dentatum. And within the, what I call the Conradina and allies section of the phylogeny, there's one little hot spot right here with Conradina etini and Signiflora. We know that because they do form hybrids currently, and they probably formed hybrids in the ancient past as well. So that's not surprising that that would have a bit of a higher score. But then the next highest scoring node in the RI index was right here at the base, the most recent common ancestor of those 3 Clinopodium species. And then we look at the introgression fraction. Well, lo and behold, the highest introgression fractions that we see are amongst those 3 species in that. Georgianum is kind of sister to the rest of Conradina and these 2 monotypic genera, and doesn't really have much to do in that regard, in terms of hybridization and introgression compared to these 3 species. So this is something that is obviously interesting to keep an eye on. And I'll mention maybe a little bit more about the reasons why. I'll mention this in a second though. So moving on from that, just looking at the species tree that we have, looking at the different nodes, trying to tease apart hybridization and things like that. The next thing I wanted to try out was what sort of phylogenetic networks could there be between these different species. So, a phylogeny, strictly speaking, is a tree, strictly bifurcating, right? It's there's no real room for genetic leakiness or incomplete lineage sorting or all these other things that can kind of lead to fuzzy or clouded relationships between different species. So using this program called PhyloNet, I looked specifically at the Dicerandra tree, a subset of it, and the Conradina and allies tree as well, to find out the probability, first of all, number of putative introgression events, of reticulation events, and between who they were, right? So from what side of the tree, what percent contribution did one clade give this other clade? So what I got for this Dicerandra tree, the highest scoring likelihood was 2 reticulation events. And in this case, it was a nested reticulation. So it was a reticulation event that was then followed immediately after by another reticulation, where the annual ancestor of, so the ancestor of all of annual Dicerandra first had this reticulation event with an ancestor of all of perennial Dicerandra, which contributed about 75% of its genomic information to this ancestor right here, right? And then another reticulation event followed shortly after that, again, had a gene flow of about 58% into this ancestor. So going back to what I was looking at before, the RI index, I had mentioned that annual Dicerandra, the node that was ancestor to the entire clade, had a relatively high score. This is a potential reason. It's likely based off of ancestral range reconstructions that I've done and collaborators have done, which I'll show momentarily, that the ancestor of both annual and perennial Glycerandra co-occurred at some point during the ancient past, during the Pleistocene in particular, and that these overlapping events and these, obviously that led to some sort of gene flow, and then that these nested, these back-to-back reticulation events led to the rise of the diversity that we see in annual Glycerandra. It doesn't necessarily explain why they are the only members of the scrub mint clade that are annuals. That also can't be directly explained by polyploidy or ploidal number or anything like that. So I guess that's another little interesting thing to look at. But yeah, so this kind of corresponds, it kind of relates back to the high score that we found for the RI index and all those other things. Then when I look at the subset of the Conradina in allies tree, PhyloNet told me that there was highest likelihoods for one reticulation event, and that one reticulation event occurred, funny enough, the ancestor of that Clinopodium clade that was mentioned earlier. And who made up, what are the genetic makeups of this ancestor? Well, it's mostly this MRCA of essentially all of these Conradina plus allies, plus some ancestor of Conradina that contributed about 41% of its genome to this ancestor of this clade too. So I guess what we can say, not definitively, but with some confidence, is that this grouping of Clinopodium here, although currently not readily forming hybrids with each other, or with, well, actually, I take that back. Clinopodium ashii readily forms hybrids with Conradina brevifolia. So these intergeneric hybrids kind of lead us to some sort of confidence that, yes, there might be some sort of Conradina genetic signal in the ancient history of this Clinopodium clade. So again, kind of, it's nice to see how one metric then kind of corroborates another metric and helps kind of give us confidence in the evolutionary history of this group, right? So as you can tell, hybridization has had a pretty strong role in shaping all the members of the species, of this clade, but in particular in Dicerandra and in this Clinopodium. So moving on from the less phylogenetics heavy aspect of this, when you look at a species richness map of scrub mints across the Southeast, there's two areas that instantly pop out to you: the Lake Wales Ridge and the Apalachicola area, right? And so these are both areas that are described in literature as being well-known endemism hotspots and well-known biogeographic sort of regions, right? So this then led me to want to answer some questions regarding biogeography of the group. So following Saurian Weekly 2001, I assigned these different biogeographic areas for the clade: Atlantic Coastal Ridge, Lake Wales Ridge, the Apalachicola River area, the Panhandle, the Altamaha River / the southeastern Georgia area, and the Cumberland Plateau, because it's a well-described refugia. And this is what I got. So this is using DIVA, which is a parsimony-based method for reconstructing ancestral areas, because it performed better than any of the other models that were used. And so, so dated phylogeny here, here's the dates, here's the legend, here's Pleistocene, Pliocene. The ancestor of all of scrub mints was likely an Apalachicola species, right? And it's from there, so this red, wherever you see red, that's Apalachicola. It's likely from there that these mints then colonize adjacent neighboring areas. So it's likely that this ancestor of the scrub mint clade was probably some sort of component of these fire-dependent wiregrass and sandhill longleaf pine savannas that were that covered the North American coastal plain during this late Pliocene into early Pleistocene period. Moving on to the ancestor of perennial Dicerandra. So this, wait, you can't really see it. So it's likely that from the Apalachicola, an ancestor then colonized the Apalachicola and Georgia area, sorry, the Apalachicola, Lake Wells Ridge, and Georgia area, kind of like right when this landmass was beginning to emerge from the ocean. And then from there, from the Lake Wells Ridge, there were subsequent dispersals to the Atlantic coastal ridge for the Thinicola and the Immaculata species. This is pretty, I guess, to be expected. So there's the eastern and western hypothesis as to the origin of most scrub species. And this kind of falls in line with the idea that most scrub species have eastern affinities, not so much western or southwestern affinities. For annual Dicerandra, we were able to reconstruct with pretty high support that this ancestor, probably from the Apalachicola region, was colonized the Georgia Altamaha region, and then from there either went back to the Apalachicola during less optimal climatic periods, because the Apalachicola is an area that's well described as being a climatic refugia, is more stable in climate during glacial maximal periods of the Pleistocene, but perhaps during interglacial periods was then expanding its range back out into that area. So that's where we have either panhandles, Georgia and panhandle, or just Georgia species here. All right. So in the case of the Conradina plus allies, again, very similar story. Apalachicola is reconstructed as having as being the ancestral region for that ancestor for that clade. But then looking at Conradina specifically, so like I mentioned before, Conradina verticillata is that one endemic to the Cumberland Plateau. It's kind of a funky story, right? It's a well-recognized refugia for North America, for these coastal plain endemics. It's likely that during some sort of interglacial period, this ancestor of all of Conradina was able to either disperse or expand its range so far northward that it was able to occur in the Cumberland Plateau. And then perhaps it stayed there during glacial maximal periods, because this area was, again, more climatically stable than surrounding areas. But then subsequently, this Apalachicola ancestor then distributed elsewhere. So it either distributed into this Altamaha, Northeast Florida, Georgia area, or when those landmasses started appearing to the Lake Wells Ridge and the Atlantic coastal ridges. All right. So timing has a lot to do with this. And I think this is probably one of the more interesting things about diversification in this group. So using a diversification rate shift analysis for the entire scrubments, there was an uptick in diversification rates somewhere about 1.4 million years ago. 1.5, 1.4, 1.3 million years ago. And here on the tree, it's more or less about there. And this corresponds almost perfectly with an area of time in the Pleistocene called the mid-Pleistocene climatic optimum, or transition. So what happened during this period? Before this period, glacial and interglacial periods were a little bit less intense, right? They were of shorter duration, but as soon as the mid-Pleistocene transition occurs, we see significantly longer interglacial and glacial maximal periods. So we see kind of like an expansion of xeric habitat during glacial maximal periods in the Southeast during this period of time. This increase in aridity, this increase in suitable habitat as well, because obviously sea levels are dropping lower now as well, so you're exposing a lot more land, a lot more sandy land to dry, to the air, kind of really allowed a lot of these already xeric adapted species, not just scrub mints, but other xeric adapted mints, to colonize, both to be able to effectively, colonize new nascent land masses like the Lake Wales Ridge, like the Atlantic Coastal Ridge, but also it gave them an advantage in older ecosystems as well, because they already went into these, drying out sandhill habitats with an adaptation for being able to deal with increased aridity as well. All right. So it seems that this increase in aridity and this increase in glacial cycling help drive diversification. So just to kind of like round this all out with these methods, because biogeographic analyses are a lot like, they can be a little bit rigid in terms of they just give us a one area, one pinpoint in time, right? What we know for a fact that the Pleistocene was a very dramatic climatic period, big changes in weather and in available land mass. So using a combination of bioclimatic variables, being able to and collecting those for extant species, we're then able to reconstruct ancestral suitability at each node across the phylogeny, and then be able to project that onto two analog scenarios during the Pleistocene, a glacial maximal period and an interglacial period, just so that we could get a vague sense of where on the landscape, where on a map, these species might have had their highest suitable habitat. So that is one thing I will say. When I'm showing a map, I'm not showing you where the species occurred. I'm showing you simply where species suitability occurred. Whether or not the species can distribute to those areas is, completely dependent on biotic factors. And so for this is the results for the ancestor of the entire scrub mint clade, which predates the Pleistocene by a little bit, but there was still a similar amount of glacial cycling. We know for a fact that Florida was mostly underwater at that time, but during glacial maximal periods, a lot of it was exposed. But I guess what you can see is there's a generalized suitable habitat essentially across the southeastern part of the coastal plain in what would be the peninsula, and in this area of the Piedmont in north central Georgia. During glacial maximal periods, unsurprisingly, distributions suitable habitat is shifted much further south, and this very this will appear again and again. This sort of like hook arrangement around this enlarged Florida platform when sea levels were much lower was likely where they were occurring, right? Similar patterns were also uncovered for this this putative range for the most recent common ancestor fall of Dicerandra. There, again, similar patterns here. There seems to be like some sort of hotspot over here at the base of the Piedmont. There's a few other hotspots here along the peninsula. Again, take this with a grain of salt because these analogs don't necessarily show the map as it was back then, but it'll at least show us suitable habitat during these analog periods, right? And then when we look at Dicerandra annuals and Dicerandra perennials, very similar sort of things show up, right? Granted, we have much higher suitability for Dicerandra annuals in this Piedmont area and north central Florida. We see lower suitability in those same areas, but we still see suitability, right? But then, these glacial maximal periods, the suitable habitat is very, very close right now, especially along the enlarged Florida peninsula, right? So what essentially this gives us a bit of a context for is these two ancestors, this ancestor for perennials and this ancestor for annuals, likely had or occupied similar ecological niches, particularly during glacial maximal periods of the Pleistocene. So it's not that crazy to think that even if they did come into contact, they were slightly exchanging some sort of genetic information, right? They were either hybridizing or just crossing either between species or even intergenerically, like we saw in the case of Conradina and Glenpodia. So when we look at locality data that we have for all of these different species, and we have our phylogeny. And I was, more or less just interested in kind of seeing, just just for curiosity's sake, what sort of what endemism hotspots appear on the map for scrub mints, and what kind of endemism are we seeing, right? So in the case of so there's several different kinds of endemism. There's I'll go over the two main ones, even though paleo is not here, because there's no paleo cells on here, surprisingly. There's neo and paleo endemism. So neo endemism essentially implies that you have a lot of closely related plants occurring more or less within the same area, right? The branches are usually shorter. They're more related to each other than you would expect by random chance. Paleoendemism, the exact opposite. You have a lot of these related species in the same area, but the branches are significantly longer, meaning that they speciate or split from each other much further in the biological past. Mixed endemism, as the name might imply, it's a mix of both neo and paleoendemism at play. And then super endemism is a kind of mixed endemism that passes a certain statistical threshold to distinguish it as such. I'm not too crazy about super endemism in the context of this, because there just aren't enough species to really make that sort of assumption. So the one cell of super endemism that was found was, not surprisingly, I guess, was this area that roughly corresponds to Edenaya State Park, where there are several different kind of range intersections between species of Conradina, some Dicerandra, and some Plinopodiums. So that makes sense. But I guess for our general interest, I really was wanting to know, I was expecting, for instance, Apalachicola to be paleoendemism. Not the case. Not statistically significant in any sort of endemism class. But the Lake Wales Ridge, at least using this clade as a model, was uncovered in several different areas as an area of mixed endemism. So there's both younger, diversified in situ clades that occupy the Lake Wales Ridge, while at the same time, also there's a higher than expected by chance abundance of older, more distantly, more anciently diverged species in the same habitat. So I guess in this role, at least looking at this clade, the Lake Wales Ridge could essentially be inferred to be both a nursery and a museum of biodiversity, right? There's both in situ diversification of these plants, and there's older lineages that come down that have already speciated, that came down here when suitable habitat arose. And then I guess this actually wasn't surprising at all, was the presence of mixed and neo-endemism along the Atlantic coastal ridge. The Atlantic coastal ridge, as a lot of you might know, is probably one of the younger ridges in Florida, right? It's mostly formed, at least all the different ridges that make it up are about 200,000 years old, right? And so I guess in the case of this mixed endemism is a little bit more foggy, but in the case of neo-endemism, the neo-endemism cells that we found directly correlate to this idea that, like, yes, this new landmass gave rise to these plants that were able to diversify in situ. They are all relatively closely related to one another, and as a result, they were labeled as such, as being a neo-endemic hotspot. And this holds up across other plant clades as well. I know Weekly has several different papers that talk about these Atlantic coastal ridge endemics and how recently diverged they are from their most closely related relatives that occur either in the peninsula or further north. So yeah, just to wrap everything up, hybridization likely did drive diversification in Dicerandra. Ancestral ranges, the reconstruction support, at least during several different times of the Pleistocene, perhaps larger ranges or expanded suitability. And they also support biogeographic the biogeographic analyses. And using this data set, we're also able to uncover these endemism hotspots along the Atlantic coastal ridge and the Lake Wales Ridge. And with that, I'd just like to thank everyone responsible for this, and shout out to FIU as well. I forgot, I guess that one logo is not there, but I'd like to take questions now, if you have any.
Yeah, thank her on Zoom.
Thank you. That was fantastic. You did a ton of work. With regard to the first conclusion on your last slide about hybridization helped the scrubment clade diversify, obviously you detected it in the extant species, but you told us there's no fossils and it's really hard to know what was around before.
Right.
Do we know much about the likelihood that hybridization causes species extinction in
there's
plants
hybrid
broadly?
depression.
And can we detect that? Like, is that a real thing?
It would be an amazing thing to be able to detect the number of times that extinction rates increased in the clade. Granted, that would, and then directly, to be able to answer that, I would need fossil. I would need some sort of geological evidence or something to be able to directly say like, oh, so I'll show you one area of the tree. So, see how these are relatively short, right? These branches are relatively short. These branches are relatively short. These are relatively short. There's some really long branch lengths here. Both subtending stachydioma, Pyloblephus, and this Clinopodium clade. Isabel San Martin in Royal Botanic Gardens, Spain, she wrote that these, she calls them spider leg branches, are usually an indication of some sort of ancient extinction event. I guess I forget exactly what sort of science or what sort of literature she cites, but it would indicate to me that there clearly has been extinction. Plenty. I mean, the fact that so many of these extant species are currently facing extinction during this quote unquote interglacial period would indicate to me that all it would take is an interglacial period to run a little bit longer than expected for some of these plants stuck in their little refugia to finally die, right? It's obviously, we're not looking at biotic directly interactions like what sort of insects, animals, what have you might have had a deleterious effect on some of these species persisting longer, just forcing their extinction. But just by looking at the tree, there are instances, and we're not. I could increase sampling, but in terms of described species, and this is a relatively well-studied group, it is interesting to me that there are these very long branches that go back. In the case of Georgianum, almost 3.5 million years. Separates it from its closest relative when a lot of its closest relatives were, only 1.5 million years off from each other. So that's something to think about. Yeah, for sure. Yeah. It's hard to tell though without a good fossil record. And that's why a lot of what I have to do is not hand wavy per se, but I have to utilize methods that can help me kind of tease apart a natural history of a group that I sadly don't have the full picture for. Yeah.
So I was curious with your potential habitat distribution maps. Was I just dreaming, or did it sort of look like a lot of potential habitat in Cuba and then the Yucatan and
so
going around?
That's the one thing I guess I mentioned was just because there is a projection for suitable habitat doesn't necessarily mean that the species occurred.
So we have no evidence of any of
No
this
evidence
in
of that.
any of
I am
Cuba.
I'm just reconstructing the
that's fine. I'm just
bioclimatic.
checking.
Yeah, I'm just reconstructing the bioclimatic niche, and then projecting that onto G space, so geographic space. Right. It's interesting. There's other southeastern mint genera that are also kind of very either narrowly endemic to the southeast. it seems to be a recurring theme for a lot of these Nepentioid mints. It has something to do with how they evolved in the Miocene in North America, how they were already predisposed to preferring these sandy, slightly xeric to occasionally mesic sort of habitats. I feel like they were, I mean, you know, they were obviously at some point likely more widely distributed than they are now. But I guess in the case of the ones that we see, the extant species that we have today, and particularly these Dicerandra perennials, is they're kind of hiding out. They're just in their refugia. They are, this is like the last little bit of suitable habitat they have given the current climatic conditions, right? And substrate conditions. So I guess I don't know if I answered exactly your question, but right. All those others. It might have something. Right. Right. Yeah. And you know, a lot of times I guess what I always like to go back to is the relatively poor dispersal capabilities that they have now. So most of these they produce nutlets, these schizocarps, and they're really their only main form of dispersal are these small mucilaginous glands that are on the outside of the seed that allow them to get carried down relatively short distances during heavy rainfall events, right? So when it comes to colonization, they're not necessarily the most adept. They don't have any sort of, they're not being eaten by birds and then pooped out elsewhere. They're not being moved by mammals because they don't produce large fruits at all, right? So that is another interesting question, right? I would be really interested in seeing how biotic interactions help them get around and help them get to the places that they're currently at, right? Because right now you look at the Dicerandra perennials and they're in such disjunct little pockets across either. Yeah.
On the east coast,
That's right.
you know, for something that can only sort of creep along and so
Right. Exactly. And that then goes back to the questions I had before of, you know, just how like looking at this biogeographic tree, the analyses essentially just kind of said, you know what, I give up. The ancestral area for these guys is either the Lake Wales or the Atlantic Coastal Ridge, even though we know for a fact the Atlantic Coastal Ridge didn't exist 2 million years ago, right? It's just one of the side effects of the method. Bits of it, right. Definitely bits of it. Of older, yeah, of older, right, exactly. So there, you know, it's, but the phylogenetic structure of the clade is not based off of geography at all. It's just this kind of like, it indicates that there was mixing of some sort on and off during these different glacial maximal periods when they were likely all kind of expanding their ranges across the expanded Florida peninsula. Yes, yeah. And they said, you know, I think at this point, all the Dicerandras that exist have been at least been found in some regard, right? I guess the one thing I did want to say is the most recently described Dicerandra fumella, and that was originally just thought to be you know, an ecotype of linearifolia. And looking at genetic information microsats, it clearly is its own species. So there might be things we, you know, again, that's why we need population level data to be able to see what sort of intraspecies and interpopulational genetic dynamics are going on, right? You know, there's some work on annual Dicerandra regarding levels of homozygosity and heterozygosity in these different loci. And the annuals are pretty surprisingly heterozygous for a lot of different things. So it means that they're not particularly inbred and they've been out crossing between populations relatively well. But then again, they have broader distributions. So that might definitely have an effect.
I was surprised that the Ashes calamint was all the way up in Georgia. Here we're
Yeah, that's according to what's it called?
Yeah, exactly. Yeah. We're interested in it here because it has its own B. So like, well, what's it doing up there? Is it does it appear to be pretty different genetically? Does it have its own B up there?
It is great. That's a great question. So I think part of the reason why this is so interesting, why this little plenipodium play that showed up is so interesting is these are relatively understudied in when it comes to scrub mints. You know, Dicerangia and then Conradina get most of the attention. I don't even know if these distinct different, these disjunct populations are all Ashii. They definitely seem very similar. They definitely corroborate that when you look at herbarium specimens of them. Genetically, I wasn't able to sample all three of these populations. I was only able to sample the Lake Wells Ridge population. But that's definitely something, I mean, just like with Coccineum, which has a similarly very weird disjunct population. There's like over here, it's over here, and it's over here, right? I forgot. I've talked to Alan Weekly about this before too, because there's a few other plants that kind of show this weird disjunct, and they're not necessarily rare, but they definitely are separated by some distance. I just think there needs to be more population genetic work on all of these, right? Georgianum forms a pretty cohesive range, and it is pretty phenotypically consistent across that range as well. But I know for a fact that habitat types along here, there's so many different riverbeds and watersheds in this area too, and this is a relatively forested area. It's very different than where I was seeing Ashii here in the middle of the scrub, right? So whoever wants to work on that, that'd be a great project.
So I'll maybe take the last question. So it looks like hybridization played a pretty big role in the evolutionary history of several of these. And for those that don't know, mints are always hybridizing with each other. And to the extent that we have a strategy here, it's keep them apart because we don't want them to hybridize. But I'm curious, like what kind of level of evidence you would want to see before we should shift that strategy of these species are meant to hybridize, and if they're going to persist, we should hybridize them.
Well,
or is that too out there?
No, that instantly makes me think of Modesta, right? It's a species that's projected to have next to nothing in terms of suitable habitat by 2080. It is clearly nested within this clade of other perennial Dicerandras. It'd be important to look at populational levels of inbreeding, homozygosity again. I'm not going to delve too much into the bioethical dilemmas of allowing things to hybridize and then infiltrate natural areas, right? But I guess if you use that logic, if only 100,000 years ago, sorry, maybe even more recently, maybe 20,000 years ago, during the last glacial maximum, 25, they were all kind of doing their thing and hybridizing with each other. Maybe that is an avenue to explore for increasing plant vigor, at least in that most critically vulnerable species, right? But
We don't need to advocate for that today.
not advocating for that by any means. Just it's a good common garden question to experiment to try out. So
All right. Well, let's thank Andre again.